Welcome back, future cardiovascular specialists. I'm Dr. Muzahid, and today we embark on what I consider the most elegant chapter in immunology. Last week, we studied innate immunity, your body's rapid response team. Today, we explore something far more sophisticated: adaptive specific host defenses. This is the immune system's special operations force, with intelligence, memory, and precision targeting. For you, as cardiotechnology students, this isn't just academic. This is the science behind why vaccines save hearts, how transplants are rejected, and why some patients develop autoimmune myocarditis. So let's begin this fascinating journey. What makes adaptive immunity so extraordinary? Two revolutionary concepts. First, specificity. Unlike innate defenses that attack general patterns, adaptive immunity creates custom made weapons for specific pathogens. Second, memory. This is the game changer. Once your body fights a pathogen, it remembers it, sometimes for life, and mounts a dramatically faster response upon re exposure. The first time your body sees a virus, it learns how to fight it. If that same virus enters again, the immune system responds faster and much more strongly. That immune memory is why we usually get chicken pox only once, and it's the basic idea behind vaccines, which help prevent serious complications like viral myocarditis. To achieve this specificity, the body deploys two specialized military branches. First, the humoral arm, commanded by B cells. Think of B cells as weapons factories. They produce and secrete antibodies, Y-shaped precision missiles that patrol the bloodstream and tissue fluids, neutralizing invaders outside our cells. Second, the cellular arm commanded by T cells. These are the special forces. They don't secrete antibodies, they directly inspect and, if necessary, eliminate our own cells that have been hijacked. Example, those cells infected with viruses that could cause myocarditis or even cancerous cells. Now, what triggers this sophisticated system? Antigens. These are the molecular wanted posters. They are specific structures, often proteins, on a pathogen surface that scream foreign to our immune system. But here's a crucial detail: immune cells don't recognize the whole antigen. They recognize small, specific regions called epitopes. A single antigen, like a bacterial flagellum, can have dozens of different epitopes. Some molecules are too small to be antigenic alone. We call these haptens. Understanding antigen structure is key to designing diagnostic tests and vaccines. This leads us to a fundamental question: how do T cells hidden within tissues see their targets? They can't just grab free-floating antigens. The answer is one of immunology's most elegant systems: the major histocompatibility complex, MHC. Think of MHC molecules as a universal display stands on the surface of cells. MHC class 1 is on almost every cell in your body. It constantly samples the proteins being made inside that cell and displays the fragments. It's the cell's status report: I'm healthy, or I'm infected with a virus. MHC class 2 is different. It's only on special cells called antigen presenting cells. Their job is to gobble up invaders, chop them up, and display the pieces to sound the alarm. This presentation is the non-negotiable first step to activating the adaptive army. So who are these professional presenters? The elite trio: dendritic cells, macrophages, and B cells. Dendritic cells are the master coordinators. They're like the intelligence officers who gather evidence from infection sites and travel to lymph nodes to brief the T cell troops. Macrophages are the frontline soldiers that also act as battlefield reporters. B cells are unique. They use their specific receptors to grab antigens with incredible efficiency before presenting them. It's like having a detective who only collects very specific evidence. And sometimes dendritic cells perform cross presentation, a clever trick to alert cytotoxic T cells about threats they didn't directly encounter. This system ensures no pathogen goes unnoticed. Look at this figure. Let's keep this simple. A dendritic cell acts like a security guard. It engulfs a bacterium by phagocytosis and traps it inside a phagosome. Lysosomes then fuse with it, forming a phagolysosome where enzymes break the bacterium into antigens. The most important antigen fragments are loaded onto MHC class II molecules and moved to the cell surface. When a T cell recognizes this antigen MHC2 complex, it becomes activated, starting the adaptive immune response. Let's follow a T cell's harrowing origin story. Born in the bone marrow, immature T cells migrate to the thymus for an education so rigorous it makes any medical school look easy. This is thymic selection. First, positive selection. Can your T cell receptor, TCR, even see our own MHC molecules? If not, you're useless. Apoptosis. Then, negative selection. Do you react too strongly to our own self proteins displayed on MHC? If yes, you're dangerous. Apoptosis. Over 98% of developing T cells fail and die. The few survivors are the perfect balance. They see MHC, but ignore self. This establishes central tolerance, the body's primary safeguard against autoimmune diseases that could target the heart. It's a ruthless but necessary quality control. The T cell's weapon is its T cell receptor, TCR. It looks like one arm of an antibody, but it's permanently anchored to the cell membrane and has one critical restriction. It can only see antigen fragments cradled within an MHC molecule. The genius lies in its diversity. How do we get millions of unique TCRs from only about 25,000 genes? Through a process called VDJ recombination. During development, gene segments are randomly shuffled, like mixing and matching Lego blocks, to create a unique TCR for each T cell clone. This genetic lottery ensures our T cell army has a receptor for almost any possible pathogen peptide, a cornerstone of immunological readiness. Graduates from the thymus wear one of two badges, CD4 or CD8. This isn't just decoration, it dictates their entire career. CD4 plus helper T cells are the generals and diplomats. They only talk to professional APCs with MHC2, and their power lies in the cytokines they secrete. Chemical commands that activate B cells, boost macrophages, and direct the overall immune response. 8 plus cytotoxic T cells, CTLs, are the assassins. They inspect any cell in the body with MHC1. If they see a viral peptide displayed, they deliver a lethal kiss of perforin and granzymes, forcing the infected cell to commit apoptosis. In cardiology, helper T cell imbalances can drive inflammatory damage, while CTLs are crucial for fighting viral heart infections, but are also the enemy in heart transplant rejection. Helper T cells aren't a single entity, they're a council of specialists. The cytokines present when a naive helper T cell is activated determine its fate. If it's a viral infection, signals push it to become a T81 cell, a commander for cell-mediated warfare, releasing IFNG to turbocharge macrophages. A parasitic worm that induces T82 cells, which secrete IL4 and IL5 to orchestrate an antibody and eosinophil attack. Need defense at the gut or lung lining? Th17 cells recruit neutrophils. This precise division of labor is beautiful, but when it goes wrong, like excessive Th2 activity, it contributes to allergic asthma, a major comorbidity in cardiac patients. Precision in immunology means everything. All right, class, let's go through this simply. A dendritic cell, which is an antigen-presenting cell, displays a pathogen fragment on MHC class II. A naive helper T cell recognizes this antigen using its T cell receptor with help from the CD4 molecule. Once this binding happens, the helper T cell becomes activated. It then undergoes clonal proliferation, meaning it makes many copies of itself. These cells differentiate into different types, such as Th1 cells, Th2 cells, and memory helper T cells, each with specific roles in coordinating the immune response. So, antigen presentation leads to activation, multiplication, and specialization of helper T cells. Now, watch the assassin at work. The cytotoxic T cell identifies its target, a cardiomyocyte silently producing virus. Step 1, TCR lock-in with the viral peptide on MHC1. Step 2, it forms a tight immunological synapse, a lethal embrace. Step 3, the kill shot. It releases perforin, punching holes in the target's membrane. Through these holes flow granzymes. Step 4, inside, granzymes trigger an enzymatic cascade, activating caspases that methodically disassemble the cell from within. Apoptosis. This is a clean, programmed death. The cell shrinks and is phagocytosed with the virus inside, preventing its release. This is how we clear viral infections from the heart muscle, a critical process in recovering from myocarditis. This diagram shows how a cytotoxic T cell kills an infected cell. An infected cell displays a pathogen antigen on its surface using MHC class I. A naive CD8 plus cytotoxic T cell recognizes this antigen through its T cell receptor and CD8 molecule, which activates the T cell. Once activated, the cytotoxic T cell releases perforin and granzymes. Perforin forms pores, allowing granzymes to enter the infected cell and trigger apoptosis, or controlled cell death. This is how the immune system removes infected cells without harming nearby healthy cells. Now, let's talk about a dangerous flaw in the system, a hijacking, super antigens. These are toxins produced by bacteria like Staphylococcus aureus. They don't play by the rules. Instead of fitting into the specific TCR binding site, they act as a molecular bridge, gluing the MHC2 on an APC to the TCR on many T cells, regardless of their specificity. Imagine flipping the master switch for 20% of your entire T cell army at once. The result is a catastrophic cytokine storm. Systemic inflammation, vasodilation, plummeting blood pressure, shock. This is the pathophysiology of toxic shock syndrome, a life-threatening emergency that can cause acute cardiac failure. It's a stark reminder of the destructive power of an unregulated immune response. While T cells train in the thymus, B cells undergo their education entirely within the bone marrow. Their weapon is the B cell receptor, BCR, which is essentially a membrane-bound antibody. They too generate incredible diversity through VDJ recombination. But their education has a similar critical goal. Eliminate the self-reactive ones, B cells that bind too strongly to self-antigens in the marrow are given a chance to edit their receptor. If they can't, they are deleted or rendered silent. This prevents the production of autoantibodies that could attack, for instance, heart tissue. The survivors are our naive B cell repertoire, ready to respond but not against ourselves. Not all B cell activation is equal. There are two pathways. And the difference is crucial for vaccine design. For most antigens, especially proteins, B cells need licensing from a helper T cell. This T-dependent activation is a conversation. The B cell shows the antigen fragment, the T cell recognizes it and provides activating signals. This leads to a high-quality response with memory and class switching to immunoglobulin G. Other antigens, like the sugary capsules of some bacteria, have repeating structures that can massively cross-link BCRs. This T-independent activation is simpler but weaker. Mostly IMG, no memory. This is why infants don't respond well to plain polysaccharide vaccines. The brilliant solution? Conjugate vaccines, where we chemically link the sugar to a protein, artificially making it T-dependent and conferring strong, lasting immunity. T-cell independent B-cell activation. Some antigens, especially polysaccharides on bacteria, have repeating epitopes. These antigens can directly bind to multiple B-cell receptors on a B cell and activate it without T-cell help. Once activated, the B cell rapidly proliferates and differentiates into plasma cells that mainly secrete IgM antibodies. This response is fast but does not produce strong immunological memory. T-cell dependent B-cell activation. In this process, a B cell first binds a protein antigen, internalizes it, and presents it on MHC class II. A CD4 plus helper T cell recognizes this antigen through its T cell receptor and provides activation signals using cytokines. This causes clonal expansion of the B cell and differentiation into plasma cells and memory B cells, producing high affinity antibodies such as IgG. So, B cells make antibodies. What do these antibodies actually do? They're multi-tools. 1. Neutralization. They can physically block a virus from entering a cell or a toxin from binding its receptor. Simple and effective. 2. Opsonization. They tag pathogens with the molecular eat me signal, the Fc region for phagocytes. 3. They call in backup by activating the complement cascade. 4. Agglutination. IgM, with its 10 arms, can clump bacteria together, making them easier to filter out. And 5. ADCC. For enemies too big to eat, like parasites, antibodies mark them for NK cells to come and inject lethal chemicals. Each antibody class is optimized for specific functions, making humoral immunity a versatile defense against bloodstream invaders that threaten the heart. This brings us to the ultimate demonstration of immunological memory, the difference between the primary and secondary antibody response. The first time you meet a pathogen, it takes a week or more to build up defenses. That's the primary response. You might get sick. But if you survive, the second time, memory B cells are waiting. They spring into action within hours. Antibody levels soar to much greater heights. The antibodies are of higher quality affinity maturation, and they are predominantly IgG, the most effective class. This is why booster shots work. This is the fundamental principle of vaccination. And for your patients, establishing a strong secondary response through vaccination is a primary strategy to prevent infections that could decompensate a fragile cardiovascular system. Let's formalize how we acquire immunity. It comes in 4 flavors. Natural active. You get sick and recover. Natural passive. Mom gives you antibodies through the placenta and breast milk. Artificial passive. We give you a shot of antibodies like after a rabies exposure. It works immediately but fades. And then there's artificial active immunity, vaccination, the crown jewel of preventative medicine. We safely mimic an infection to trick the body into generating its own long-lasting memory and protection. This is applied immunology at its finest, and its impact on public health, especially in preventing cardiovascular complications of infectious diseases, is immeasurable. Vaccines are not all made the same. We have a whole toolbox. The classic live attenuated vaccine like MMR gives a robust lifelong memory but carries a tiny risk. Inactivated vaccines like the flu shot are safer but often need boosters. Subunit vaccines like for HPV use just a piece of the pathogen. For diseases caused by toxins, we use toxoids like for tetanus. A brilliant innovation is the conjugate vaccine solving the infant response problem, and the newest star, mRNA vaccines. These aren't live viruses. They're instructions that tell our own cells to make the antigen, brilliantly stimulating both arms of immunity. Each type is a testament to our understanding of adaptive immunology, and each plays a role in protecting cardiovascular health. Let's put it all together with a case. Imagine an infant with fever, stiff neck, and a rash that doesn't blanch. Meningococcal sepsis. The culprit is a bacterium with a sugary capsule. The infant's immune system sees this capsule as a T-independent antigen. The response is weak, short-lived IgM, no memory. The infection can rage, causing septic shock that crashes the cardiovascular system. Now, the immunological solution, we take that polysaccharide and chemically weld it to a harmless protein like tetanus toxoid. This conjugate vaccine is a masterstroke. Now, when administered, B cells see the sugar, internalize the whole conjugate, and present the protein part to helper T cells. We have now engaged T-dependent immunity. The result? Strong IgG, lasting memory, and protection for the very young. This is applied immunology saving lives and hearts every single day. So, to summarize this magnificent system, adaptive immunity is a symphony of specificity and memory, conducted by MHC molecules and played by lymphocytes. B cells provide the antibody shield. T cells provide cellular surveillance and command. The first movement, the primary response, is adagio, slow and deliberate, but upon re-encounter, the secondary response is presto, fast and powerful. As future cardiotechnology professionals, this isn't abstract science. This is the framework for understanding why we vaccinate against flu and pneumonia. This explains why heart transplants are rejected and how we suppress it. This underpins why some patients develop autoimmune heart disease. You are now equipped with the knowledge of the body's most intelligent defense system. Use it to protect the heart, not just through machines, but through immunological insight. That brings us to the end of today's lecture. If you found this explanation helpful, please like, share, and subscribe to the channel. Stay curious and keep learning, because science grows with questions. If you have any doubts or questions, drop them in the comments, and I'll be happy to respond. Don't forget to follow the Modern Microbiology Academy channel for more clear and concept-based lectures. Thank you for watching.